Hello, my name is Mani Kuchru, and I am an MD-PhD candidate in the Krishnaswamy Lab. I would like to thank LMRL for the opportunity to present our new approach to high-dimensional data analysis. Biomedical data is being generated at increasingly massive scales. With increasingly high-dimensional data, it becomes important to pick resolutions at which to perform analysis. Viewing data at any one resolution will miss information present at different resolutions. Just look at this model here. Viewing all the information present here at the level of the organism will ignore vital information present at the molecular level and at the population level. For this reason, an important question in machine learning and biology is how to view and analyze increasingly massive data at multiple resolutions in a computationally efficient manner. To analyze data across multiple granularities, we created Multiscale PHATE, which is based on previous work in manifold learning via PHATE and data coarse graining via diffusion condensation. First, we create the biological manifold by computing the potential distance on our data as done by PHATE. Then we apply diffusion condensation's iterative condensation approach to learn a hierarchical tree from this learned biological manifold. By cutting this tree at different granularities, we can create visualizations at different resolutions. By cutting it at a coarse level, we see fewer points, giving a higher level summarization of the data. And if we cut the data at, or the tree at lower levels, we can see finer grained or more detailed representation of the data. In these multiscale PHATE visualizations, colors represent clusters. The size of each point denotes the number of cells that are represented in that point, or some view of the density of the data. And distances between points to know the overall similarity or differences between points. Lastly, our approach is incredibly fast and can embed 5 million cells in just over 10 minutes. This is orders of magnitude faster than any previous visualization and clustering techniques. We applied our approach to characterize 22 million blood cells measured from 168 patients hospitalized with COVID-19 at the Yale New Haven Hospital. These cells were measured with flow cytometry. Using cell type specific markers, we identified all major cell types, as can be seen here. In order to identify cell types enriched in patients that died from infection, we computed MELD mortality likelihood scores and mapped them onto our identified cellular populations. Higher mortality scores or greater enrichment in patients that died are visualized here in darker red, while cells more enriched in patients that survived are in blue. The analysis here helped us identify the granulocyte population, shown in green, are most enriched in patients that died from COVID-19. Further analysis in the granulocyte cluster identifies two subsets, eosinophils and neutrophils, and within the neutrophil population, two further subsets, CD16 high neutrophils and CD16 low neutrophils. Within the neutrophil cluster, we identified that these two neutrophil subtypes are actually significantly differentially enriched in patients that die, with CD16 high neutrophils being greatly enriched in patients that die and CD16 low neutrophils not being enriched in patients that die. We performed DREAMY or nonlinear correlation or association analysis with various markers that are found on neutrophils and found that CD16b was negatively correlated with mortality, while forward scatter or size and side scatter or complexity were positively correlated with mortality. This implies that a non-activated and immature neutrophil population plays a role in mortality. This work was a joint effort between 4 labs at Yale School of Medicine and University of Montreal. The Krishnaswamy and Wolf labs led computational tool development, and the Iwasaki and Houston labs helped inform and guide analyses on patient PBMC data and viral genomes data, respectively. Lastly, please do visit our lab website and GitHub, and please follow us on Twitter. A preprint manuscript is available on bioRxiv, as can be seen here. And please also visit our Multiscale PHATE GitHub page and download our code. Thank you very much.